Thank you for the introduction. As you mentioned, I'm Chiara Rossi, and I'm a PhD student from University of Georgia. And today I will present my project, which is about the effect of Dodine on photosynthetic efficiency and yield of peanuts. So I would like to start this presentation with a little bit of introduction. Then I will move on the objectives, the methodology. I will show you some of our results. Then I will go through the conclusion, and at the end, future research. So peanut plants are very susceptible to many soil-borne and foliar disease. And one of the most common disease here in Georgia is leaf spot, which includes early leaf spot and late leaf spot. This disease can cause many damages, including defoliation, reducing text strength, lowering pod production, and eventually also lowering yield. Since there are no cultivars with higher resistance to this disease, many fungicide programs have been developed and used in the past years. And one of the most widely used fungicides, especially in peanut crops, is chlorothalonil, which allows growers to control the intensity of this disease, so of both early and late leaf spot. However, chlorothalonil was recently banned in Europe due to its high risk to amphibians and fish. On the other hand, Dodine is an alternative fungicide with similar range of activity, but recent studies have demonstrated that it suppresses photosynthesis in some species like pecan. However, there are no studies related to the effect of Dodine on the photosynthetic activity of peanut plants. And that's why the main objectives of this project were to assess the effect of Dodine on the photosynthetic activity of peanut plants and verify the potential of Dodine as a replacement of chlorothalonil. So based on this, our hypothesis is that Dodine will not affect the photosynthetic activity or yield in peanuts negatively, and so it can be a potential replacement for chlorothalonil. So our experiment was conducted in Lang Farm, which is part of University of Georgia Research Center in Tifton, and it was conducted last year in 2020 using the cultivar Georgia 06G. For the treatment list, we used the two fungicides with two different rates. So we had chlorothalonil full rate and half rate, and then Dodine full rate and half rate. The size of our plot had a length of 40 feet and a width of 12 feet. And for the experimental design, we chose a randomized complete block with 5 replications. I will now show you a flow chart that represents all the measurements that we collected in the field. And then I will explain how we collected these measurements. So first of all, the field was planted on May 14th. And then at 32 days after planting, we collected the first physiological measurements as a baseline. After that, we started with the fungicide applications, and we kept collecting physiological measurements for a total of 7 fungicide applications and 8 physiological sampling events. At 146 days after planting, we dug the field, and lately, at 149 days after planting, we collect pods in order to create the pod maturity profile. Lastly, at 156 days after planting, we harvest and we calculate the yield. Now, before moving on the physiological parameters that we wanted to study, I want to show you the simplistic representation of a chloroplast. Chloroplasts are very important because it's where photosynthesis occurs. And photosynthesis is mostly made by 2 sets of reactions, the light reaction and the Calvin cycle. In the light reaction, pigments are important because they are responsible to intercept and absorb light that then is used to move electrons from one molecule to another one until water is oxidated and ATP is produced. And ATP is the main source of energy for the plant. In the Calvin cycle, instead, an important process is gas exchange, which is basically the exchange of CO2 between the leaf and the surrounding air. And this process is highly correlated with stomata closure or stomata opening. So based on this, one of the physiological parameters that we decided to study was gas exchange and fluorescence, which includes different parameters like net photosynthesis, stomata conductance, electron transport rate, and quantum yield of the photosystem too. All these measurements were obtained with an infrared gas analyzer that is shown in this slide. Another important study that we did was related to the pigment content, which included total chlorophyll, so chlorophyll A and B, and total carotenoid. For this analysis, we collected these leaves and placed them in 95% ethyl alcohol denatured solution for 14 days. And after that, with a spectrophotometer, we did absorbance reading at 3 different wavelengths, 470, 469, and 665 nanometers. For the pod maturity profile, instead, we collected approximately 200 pods from 5 different plants, and then we pressure washed them in order to remove the outside layer and reveal the color of the mesocarp. We then placed all the pods in a pinna profile board, and the position of the pods in the board, especially when they reach that slope line in the black class, that represents the approximate time needed for the plants to reach maturity and so to be inverted. Lastly, we calculate yield at harvest, and we adjusted 7% of moisture. For the statistical analysis, we use analysis of variance and to test a 5% probability with JUMP software. Moving now on the results, this first graph represents the total chlorophyll content, so chlorophyll A and B, based on days after planting and divided for the 4 fungicide treatment. So as we can see, we had an increase of total chlorophyll content throughout the season, but this was mostly based on the growing stage of the plant. In fact, we did not find any significant differences between our fungicide treatments. This second graph represents the total carotenoid content based on days after planting and divided for the 4 fungicide treatment. Also in this case, we can notice an increase in total carotenoid content throughout the season, but again, this was mostly based on the growing stage of the plant. In fact, also in this analysis, we did not find any significant differences between our treatments. And these two graphs indicate that both total carotenoids and total chlorophyll were not altered by the fungicides, but also by their rate. This third graph represents net photosynthesis based on days after planting and divided for the 4 fungicide treatments. So in this graph, we can see some variation throughout the season, but it was mostly based on the growing stage of the plant and also on weather conditions. In fact, we did not find any significant differences among our treatments. But one important thing is that net photosynthesis is highly correlated with other physiological parameters, for example, somatoconductance, electron transport rate, and actual quantum yield of the photosystem too. And since they are highly correlated, they all follow this very similar trend. In fact, also these 3 parameters were not significant difference among our treatments. This fourth graph represents the percentage of pod maturity based on the 6 different stages and divided for the 4 fungicide treatments. So since peanuts have an indeterminate growth, it can happen that in one plant we have pods with different maturity. And that's why in this graph, we can see pods located in all the stages and with different percentage. So the first stage is the white one, which is the less mature one, and it's followed by yellow 1, yellow 2, orange, and the last 2, which are brown and black. Also for this analysis, we did not find any significant differences, but one thing that I wanted to point out here is the PMI, which is the percentage of brown and black together, and it indicates the pinna maturity index. A study conducted by Diana Rowland demonstrated that a PMI of 70% is optimum for yield. And if we look at the PMI that we obtained, the chlorothalonil full rate, the first fungicide, that had a lowest PMI, all the other 3 had a PMI really close to 70%. Lastly, this graph represents the yield data that we obtain in our field, and it's divided for the 4 fungicide treatments. So although chlorothalonil full rate had a slight highest yield than the other 3 fungicides, Also, in this case, we did not find any significant differences among our treatments. So in conclusion, I know that the results that we obtain are not what we are usually looking for. What we usually want to see are significant differences among our treatments. However, for this specific project, these results are very exciting because they can accept our hypothesis that was that Dodine will not affect photosynthetic activity or yield in peanuts in a negative way, and so it could be a potential replacement for chlorothalonil. And we can confirm and accept our hypothesis mostly for two reasons. One, because Dodine actually had similar effects than chlorothalonil on the photosynthetic activity of peanut plants, but also on pod maturity profile and final yield. And secondly, since we did not find any negative effect after the Dodine application on our peanuts, this chemistry can actually be a potential replacement of chlorothalonil in the control of the intensity of leaf spot in peanut plants. However, there are for sure future research that can be done. One of them is to validate the results that we obtained last year by conducting this research again this year, and we actually already started collecting measurements. The second one is to use different cultivars with different range of resistance to leaf spot. And the third one is to conduct this experiment in different locations in order to expose the plant to different disease pressure. With that, I would like to say thank you to all the professors that participated in this project, especially my advisor, Dr. Cristiani Pilon, to all the team effort, and to Georgia Seed Development and National Peanut Board for funding this project. Thank you, and with that, I will accept any questions you might have.
Thank you, Chiara, for a very nice presentation.
Thank you.
First question's coming in now. An anonymous attendee. What was the level of leaf spot in the test? Any difference between the Dodine and chlorothalonil efficacy on leaf spot?
So, okay. So the question is if there is a difference in leaf spot between the fungicide treatment. So we actually did not take measurements on leaf spot because at the end of the season, we did not see visually differences between our treatments. So this is something that we are planning to do this year. So hopefully next year in the next conference, I can show this result. Thank you.
Okay. What was the question I have is, what was the initial reason for looking at this? I've looked at Dodine before. What led you to a project to this objective? What was I understand looking for replacements to chlorothalonil, but why a concern over Dodine?
Okay, so the question is, what is the aim of looking for a substitute to chlorothalonil? Well, first of all, chlorothalonil was recently banned in Europe because of its high risk to some animals. And second, because since it's one of the most used fungicides, especially here in the US, the demand of this fungicide is really high. So it can happen that not everyone is gonna be able to use this fungicide. And that's why we're trying to find a substitute that can be used in case chlorothalonil is not available.
Very good answer. And thank you very much, Chiara.
Thank you.
That's the time we have now.
Okay, thank you.